genomic sequence databases. And specifically, I'm going to be talking to you about Jingyu's work on duplicate detection. And so the context of this work is really trying to investigate what's inside of databases like GenBank and trying to explore the problems that might exist in that resource. And it's been observed, going back to quite a long time ago, that there are a number of potential kinds of quality issues in our bioinformatics databases, duplication being one of them and the focus of this particular study, but there are other problems in these databases, inconsistencies, inaccuracies, untimeliness, meaning that there's a lack of currency of some of the data in the databases, and of course incompleteness. And so really what we're trying to do is to recognize that these databases are not perfect, and in order to inform how we use these databases and eventually try to clean them up. And so for this particular project, we are focusing on duplication. And it's probably worth acknowledging that outside of the bioinformatics community, there's been a lot of work actually on duplicate detection. Less work in the biological database context specifically. And that the work that has existed or has been done previously has focused kind of on two particular areas. One is trying to find duplicates very quickly, and the priority for those methods is efficiency, and essentially those methods tend to assume that the sequences in the database have very high sequence identities. So they start with a sequence identity threshold and move from there. Quality-focused methods look more broadly at the data that's inside of each record in the database and have a more nuanced definition, but they tend to be a bit slower. But they're extremely important for curation of the databases. But when we've started investigating this literature, we kind of noticed that there was actually not a lot of detailed analysis of what duplicates are, even what a definition is. And you can see from the different kinds of methods that there isn't really a consensus on exactly what these duplicates are. And there are also no validated benchmarks that you can use to approach this problem. So the previous work has really used kind of a very small sample of the possible things. So what we've been trying to do is to collect a larger benchmark data set, and in the end we've pulled out over 1 million pairs of duplicates by leveraging a mapping between the UniProt databases and the nucleic acid sequence databases, and essentially going on the assumption that if there's more than one nucleotide record corresponding to a given protein record, then those two nucleotide records should be considered duplicates of each other. And in fact, we find that there are different kinds of duplicates in which this happens. So there are, of course, sequences which are almost identical, and then we also have fragments and non-significant alignments emerging in there. So what we did for this particular work was to build a supervised learning approach that takes into consideration not only the sequence similarity of a pair of records, but also a number of other kind of more qualitative aspects of the records. So we measure the similarity of the description of a pair of records and other information that's inside of the DNA record for the sequence. So we take a pair of records, we represent it in terms of these various similarity metrics according to the different aspects of the record, and then we build a machine learning algorithm. We apply a machine learning algorithm to build a supervised model of the distinction and try to make a prediction whether a pair of records is a duplicate record or a distinct record, not duplicate. And we have two paradigms in which we've done it, just a simple binary classification, and one that actually takes into consideration the different types of duplicates that we find in there, with the intuition being that you may be better off building a classifier that distinguishes between those different types. Okay, so the basic story is that if you compare against that sequence similarity thresholding strategy that most previous work has used, you actually get very poor performance with that baseline method and substantially improved performance using a proper machine learning algorithm. So whether that be naive Bayes or decision trees or SVMs, you see a substantial improvement. And the story over here on the top right is that, of course, the more data you have in general, the better you can do this. And so for different organisms with different amounts of training data, you will get different levels of performance. Of course, we also want to understand the impact of that sequence similarity metric in comparison to the more qualitative factors. And so we did an ablation study, and it turns out that just looking at sequence is not enough to solve this problem. Okay, so the basic story is that supervised learning shows substantial promise for this problem, and if you want to know more, you can come and talk to me at poster N51 later today. Thank you.